Hi Genetics 311, my name is Val Perez, and in this video I will be discussing the Watson and Crick model of DNA. Watson and Crick brought together data from previous research done on DNA to come up with their model. The structure is represented by a double-stranded anti-parallel right-handed helix. The molecule contains two sugar phosphate backbones on the outside, being held together by hydrogen bonds between the pairs of nitrogenous bases on the inside.
DNA is a polymer of linked nucleotides. These nucleotides are composed of a sugar, a base, and a phosphate. The phosphate is going to attach to the sugar at the 5' carbon, and it's what makes DNA bear its negative charge. The nitrogenous base is going to attach to the 1' carbon of the sugar. The next nucleotide bearing a phosphate on the 5' carbon is going to attach to the 3' carbon of this nucleotide. Then on the 2' carbon, we can see the hydrogen that distinguishes DNA from RNA. These are the nitrogenous bases. They bear the genetic code. There are two types of nitrogenous bases, the purines and the pyrimidine. The purines include adenine and guanine, while the pyrimidines include cytosine, thymine, only in DNA, and uracil, only on RNA. Let's discuss complementary base pairing. Complementary base pairing refers to the hydrogen bonding between the nitrogenous bases, and it is the phenomenon responsible for the double helix structure of DNA. Adenine is going to form 2 hydrogen bonds with thymine, while guanine is going to form 3 hydrogen bonds with cytosine. These are relatively weak attractions between the partial positive and partial negative charges on both of the molecules. Even though these bonds hold the double helix together, they are not the main force joining the two strands. Most of the stability of the structure is due to the interactions resulting from base stacking. Look at the basic structure of DNA. Phosphodiester bonds are going to connect the sugar phosphate backbone, where the 3' end of one nucleotide is going to bind to the 5' end of another nucleotide. We read DNA in the 5' to 3' direction. However, both of these strands are anti-parallel to each other, meaning one is read from 5' to 3' and the other one is read from 3' to 5'. The center is going to contain the hydrophobic bases, and the hydrophilic backbone is going to be on the outside. Strands of DNA form a plectonic coil that can't be pulled apart. This type of coiling of the double bond helix, where the two strands are intertwined and cannot be separated without uncoiling first. Here we can also see the major groups and the minor groups of the DNA. Previously mentioned, most of the stability of the double helix structure is due to a phenomenon called base stacking. Base stacking is going to reduce the exposure of the hydrophobic bases to the aqueous environment, and it is achieved by the bases moving closer together. The backbone is going to be tilted approximately 30 degrees, which puts the bases in a planar position, one on top of the other. This maximizes the hydrophobic interaction and the van der Waals forces between them.
Some examples of left-handed DNA I found on Twitter and the news. How did I know they were left-handed? I always imagine myself going up a set of spiral stairs, and whichever hand is on the rail is the handedness of the coil. You can also tell by which direction the angle is facing. On the first image, I laid out a line that shows you that the DNA is going to the left rather than the right. The same is true for the rest of the images. Alright, that concludes my video. Thank you for watching.